Yeah, today I will talk about some computational techniques in drug repositioning, both from the molecular point of view and from the machine learning approach. This is in the outline of my presentation, which is a compressed journey of through my PhD in Belgium and part of it also in Germany at the TU Dresden. And we start talking about the general concept of drug repositioning and what drug repositioning aims to do. And I will follow with analysis of the current status of drug repositioning and the rational approach, which is usually behind the computational techniques that can help the process of repurposing drugs. And at the end, I will explore with you the how we can fuse different kind of data in order to increase the accuracy of prediction of new indications for existing drugs. And so we will dip a little bit more into the machine learning and deep learning. So first of all, let's start with this general concept of what drug repositioning is and what drug repositioning is trying to do. And in order to do that, we have to start from the main problem, which is the negative trend of drug discovery. As you can see from the graph, starting from the 50s when the data started being recording, all the way to to now. Well, I've been recording until 2017 because it was when I was doing my PhD at the time. We've been spending more and more money and bringing less and less drugs on the market. So why is this happening? Why every year we are spending more and getting less drugs available for the therapies? Well, there are different theories about that, and I want to share with you a couple of ideas about that. One is the oil drilling theory. And according to this theory, over the time, we've been exploiting the easiest and more profitable sources of drugs. And we've been spending money on indications that were more profitable, and we left on the bottom of the barrel, let's say, the drugs that were more difficult to find, and also they were less profitable. So over the year, we will need more and more money to find and get those drugs on the market. And another theory is related with the increasing standard of care. So every time we bring a new drug on the market, we are increasing the standard of care. So we will need more and more studies, more and more data in order to make the next drug safer than the previous one. And that is impacting a lot on the total amount of money that we have to spend to bring a new entity on the market. Of course, we are also developing different techniques that can help overcoming this problem. And one of the approaches that over the years has been always explored and used more and more is the drug repurposing. Drug repurposing means using a drugs that we already have on the market or that has been rejected in one of the clinical trials in order to save time and money to get this drug on the market following a different therapeutic indication. The numbers here are quite different, but the data says that we can save up to half of the time and half of the money compared to the standard process of drug discovery, which can get to $2 billion of US dollars and up to 20 years of studies. So drug repurposing is not a new thing. It's something that we've been using over the time, and there are already a lot of drugs on the market that we have repurposed and repositioned over the years. And here I gave you a short list of some of those, some examples from drug repurposing, but there is one that I highlighted and that I wanted to spend a couple of words more because it's been a very hot case, mostly in the past, and is the thalidomide story. So thalidomide is a drug that was for the first time brought on the market after the Second World War in Germany in the late 50s, and it was used as a sedative and immediately gained a lot of interest for the ability of stop the morning sickness in pregnant women. It was quite safe, so it was tested on mice and rabbits with amazing results, and it gained immediately great popularity, until some years after, when they started associating the usage of this thalidomide, this drug, with increased birth of phocomelic children, so children with very small or even no limbs and arms and legs. The drug was immediately taken out from the market, and for a long time has been kind of taboo in pharmacology, and of course was unthinkable to use it again, this drug, since the test that they were carried, of course, didn't take into account the teratogenesis, and it was not tested on pregnant women or pregnant animal, so it was not possible to understand the full range of side effects. Later on, the teratogenic effect was connected with the anti-angiogenesis, so the ability of stopping the growth of blood vessels, and that became this side effect became actually a very strong therapeutic indication in order to stop the growth of blood vessel in tumor. So it became a very strong therapeutic tool against multiple myeloma. So something that was perceived very bad then turned out to be actually an advantage for a different indication. So that's the main concept of drug repositioning, drug repurposing. But how do we stand now in terms of drug repositioning? So this is one of the studies that I did during my PhD, and together with a group at the TU Dresden, we mapped different databases available at the moment, and we try to put together the information that we currently have about drug repurposing to understand how far we are now with this approach. So we use the drug repurposing portal, which contains almost 200 drugs that have been repurposed and available on the market. We met those drugs that are connected with the first and second indication with a list of drug target couples that has been published in 2016 on Nature. And together with that, we use the literature on PubMed to close all the connections between the missing connection between the target and indication. And in this way, we could make a network drug targets indication from all the repurposed drugs and understand how we've been so far approaching to drug repositioning. What we saw in the beginning as a first thing is that looking at which kind of disease the drugs have been repositioned, we can see that on the main diagonal, most of the green squares are actually aligning. That means that most of the drugs have been repurposed from the one disease to the exact same class of diseases. So this graph has been built using the mesh key, which is a way to characterize and express the different therapeutic indications in a medical group of diseases. And since the diagonal is the brightest and most colored place where the squares are, that means we've been repurposing most of the drugs from the same mesh indication to the same one. So, for example, most of the drugs have been repurposed from C4 to C4. That means from one kind of neoplasm to another kind of neoplasm, so the same therapeutic indication. An example is nilotinib, which is a kinase inhibitors that has been has been repurposed from leukemia to gastrointestinal stromal tumor. That falls inside the same mesh category and has been considered for us a kind of disease indication-based repositioning. Then we went forward and we look at from the target point of view, which fraction of those repurposed drugs share the same target. And 40% of all the cases that we observed are actually sharing the same drug target. This is a slightly different concept than drug repositioning, but this is called target repositioning. So the target that can be associated to a different indication is able to bring with them all the different drugs that are connected to the target and for the other indication. And that allows us to use different and more therapeutic tools for a new indication in a very fast way, because we don't have to restate the interaction between a drug and a target that's already there, that's already existing. We just have to prove the connection between an indication and a target. An example is the celecoxib, which has been first approved against rheumatoid arthritis, and then against the familial polyposis, through the connection with the same target, the COX-2, the cyclooxygenase 2. And that's a case of target repositioning, which is, again, a very big piece of all the drug repositioning cases that we analyzed. And only at the very end, at the very bottom of all this analysis, we found a few cases where the drugs were repurposed from one target and one disease to a different target and a different diseases. Why are those cases so interesting? Well, going from one target to a different one in a different disease allow us to have a higher extent of diversity, chemical diversity, therapeutic diversity, and that is very appealing, for example, for a drug discovery project, for a pharma company who invests in our research, because we are able to provide a completely different chemical entity that has not been explored already for that kind of indication or for that kind of target, and maximizing the chance of profits. And the number, I put together the number in this graph to show the disparity between all the different approaches that so far has been used for drug repurposing. Almost 60% of drugs have been repurposed inside the same disease class. 36% have been repurposed to a different disease, but to the same target, and only 5% have a different target and a different indication. So only those 5% have a new drug target interaction, which is maybe the most interesting from the academic point of view between the among all the repositioning processes. So now I'm gonna show you some approaches that have been exploring and studying in order to make this drug target interaction prediction more possible to happen. So I summarize here the way how the three different kinds of repositioning approaches that I showed you before can be expressed in the drug target and indication network. So starting from the left of the graph, the basic concept is that therapeutic action can be expressed and can be shown by the interaction between a drug in green, a target in blue, and the different action, the way how the drug can modify the action of a target can then be translated in a therapeutic indication in yellow, and a lot of different side effects in red. This concept is called polypharmacology, the ability of a drug to be able to be involved in so many different therapeutic indication and biological effects through the connection with different target is the basic concept of the rational drug repurposing. So using a rational approach, how can we start from a drug target indication and go to a different one in those three different ways that I'm showing here? One is the disease centric, the first one on the left in the gray panel, which is actually related with the 59% of the cases that I showed you the first slide, where a drug is repurposed to a different indication just because of the similarity between the side effect and of the drug and the therapeutic indication, the first therapeutic indication of the same drug. So in that case, I don't really need to build a new interaction between a drug and a target for the second indication. I will just go from indication 1 to indication 2 if they are close enough to suggest me that the drug can be a good candidate to be repurposed. And the second approach is the target-centric, which is the one in the middle and related to the 36% of the cases that I showed you previously. In this case, I don't need an interaction between a new interaction between the drug and the target, but the only thing I need is to be able to repurpose the target from the indication 1 to indication 2. So that's why it can also be called target repurposing. It's a little bit more complex because it needs extra information, not just similarity between indication, but a biological connection between the involvement of the target, between the target being involved in the indication 1 and indication 2. And then we have the third case, which is the drug-centric, is the one that I'm trying to push a little bit more on, and is the one that also requires more information because it needs to build a new interaction between a drug and a new target that is connected with a second indication. And that's why only 5% of the cases that we analyzed in our database is actually coming from this approach. Now, focusing only on the third approach, the drug-centric, so the one that allows us to build a new drug-target interaction for drug repurposing. There are many techniques that can be used, computational techniques that can be used and can help this rational approach, and that can explore the polypharmacology of all the drugs that we have in order to repurpose and to suggest new candidates for a different therapeutic indication. I divided those approaches in two big classes, which is usually like the canonical way of separating those techniques. One is the ligand-based. The ligand-based approaches are the one that take into account all the different biologically effective molecules for a certain target, and making a study on all those drugs is possible to build maps connected with biological activity that express a correlation between the activity and specific physicochemical characteristics of all those drugs. An example is, of course, the 3D QSAR or the pharmacophore modeling. However, those techniques need a lot of different biological active compounds for the same target that have to be superposed. And sometimes it's difficult with those techniques to get over the chemical class of specific drugs that will always bias the prediction toward a specific class of drugs. And then there are the structure-based techniques. The structure-based techniques they involve the structure of a target in order to carry on the prediction. And I subdivided the structure-based techniques in two different kind of techniques, the target-based and the interaction-based. The target-based, for example, the docking or binding site similarity, is strictly connected with the three-dimensional structure of the protein that can be available from crystallography or using different kind of modeling techniques. And the second category of techniques is the interaction based, and this class of techniques doesn't necessarily is not necessarily connected with the three-dimensional structure of the target, but only with the interaction profile and the physical-chemical interaction that connect and link the ligand with the amino acid in the target, without taking strictly into account the sequence of the target, the three-dimensional structure of the residues of the target, and even nor the one of the ligand, but only the environment, the interaction environment between the two. So those three different groups that I showed you can be summarized in this way. The structure-based techniques that I showed you, so the target-based and interaction-based, can be summarized in this way, in their differences and in their similarities. So the first one, using a same drug to find predict the interaction with different targets, is the base of the reverse docking. So when I try to fit the same drug into different targets to find the new targets for the same drug, most of the time I use reverse docking. So I try to fit the drug inside the binding site of the protein. And in this case, for example, it's shown how the same drug, the brivudin, can fit both in the HSP27 and thymidine kinase binding site because of the interaction with those two residues, the two phenylalanine that are placed in the same, more or less in the same way, in the two different binding sites. Another way is the canonical docking, is when from the same target we are trying to find more ligands than the one that are currently available. And in this case, you can see from the same receptor, the 5-HT2C protein, for the receptor 5-HT2C, we can fit inside two different ligands, the ritanserin and ergotamine, and both of them, for both of them, is important the interaction with the aspartate 134. So this is a way to explain how different ligands can actually fit into the same binding site. For the interaction profile similarity method, like you can see here, it's possible to start from a coupled drug target and land into another coupled drug target that are completely unrelated with each other, and they share only the physical-chemical way of building the interaction inside the binding site. So in this case, there are two different couples, VEGFR2 pazopanib, which is an anti-cancer kinase inhibitors, and another kinase inhibitor, ibrutinib, with this one of its target, TKP1. And in those cases, you can see there is no residues shared or moiety or a specific chemical moiety shared among the different entities, but it's the complex interaction profile that they can be overlapped and can be similar, and that can give us the suggestion how to repurpose a drug from one target to another. So now I'm going to show you more in the specific how this interaction profile similarity approach works, because this is the one that I've been using to carry my drug repositioning protocols. And I will show you in the specific one tool, which is called PLIP, developed and published by the group in Michael Schroeder at TU Dresden. So as I explained you before, for interaction profile similarity tools, the important thing is the profile, the interaction profile, the environment inside the binding site, that is characterizing the interaction between the drug and the target, and not a specific residues or a specific chemical atom on the ligand. So what this tool and the other tools for interaction profile similarity do is converting the physical-chemical information of the interaction into numbers and mathematical entities, so angles and numbers, that can be used to easily make a similarity assessment of the different environment. So each bond is translated into a category, a physicochemical category. Can be H bond, can be salt bridge, water bridge, and so on. And the length of the bond, the angles between the different bonds is also important and is described, and is going to give at the end a picture, an overall picture on how the drug and the targets are behaving in order to interact. So we use this tool in order to find, to fish among the existing drugs some new inhibitors of B-cell activation that could be used in order to suggest a new tool against autoimmunity, since many of autoimmune diseases are actually carried by B-cell overactivation. So we started from a pool of drugs, drug target, 22 kinases that we selected for the ability of overactivating different markers that are typical in a B-cell, expressed on the B-cell surface. And using those 22 kinases, we went on PDB and we found their structure. And we carried this virtual screening with this virtual screening pipeline, and we selected 110 drugs that we could then test. And after the in vitro testing, we had 20 out of 120 drugs that were available, actually able to recognize and inhibit the kinase target. And one of those 20 new inhibitors were actually able to inhibit the cell model, so the B cell that we were overactivating in vitro. So this is the graph to show the physical interaction between the two of the best 20 of our inhibitors. And what we were curious was, is it actually this interaction profile similarity working? And we went looking at those two among our best candidates, and we saw that actually the interaction profile between the first target and the new target were kind of similar. There was no similarity for in terms of sequence or in terms of chemistry of the ligand, but there was a similarity in the behavior, how those entities interact. Then we wanted to see if this approach was actually useful, and so we looked at the kinase inhibitors that we already had on the databases that were able to inhibit those new targets. And what we saw is that all the inhibitors, so the inhibitors that were already available on the targets, were on the database, were all chemically connected, as you can see on the top left part of the graph, and they all share a huge amount of same targets, so that increases the chance of side effects. While the one that we fished using the interaction profile similarity were actually way more diverse. And this chemical diversity can be useful in order to avoid side effects and give a little bit more specificity. The problem is the availability of the structural data. In order to have interaction profile similarity pipeline, you need to have a model or a crystal of a target together with a ligand, and more of those you have, and better is going to be the outcome. And for the 22 kinases that we start in the beginning, only 10 of them on 2018, when we started doing this job, were actually available. The problem is exactly the availability of those structural information. And in fact, for all the drugs that we analyzed in the when we did the current status of drug repositioning work, none of them was able to be repurposed using interaction profile similarity pipeline, because there were not two, there was no structural data about the first and the second indication for any of the drug. So this is one of the big limitations, of course, of the structural drug repurposing. And if we want to be optimistic, we know that the PDB database is growing faster and faster. So we expect the number of structures to reach a very good point in 10 years if we extrapolate the data that we have so far. But of course, we can help this process using homology modeling and machine learning. So I had to insert this, of course, because it happened just yesterday, the news that using machine learning and deep learning, people from DeepMinds were able to resolve the structure of many different proteins and explain their folding behavior only using machine learning and not using crystallography. So the future looks bright. We know that we have the chances to increase our structures in one way or another more and more. But we can also use at the moment the machine learning. So that's the conclusion of my presentation. The last part of my PhD was trying to improve and increase the interaction, the drug target interaction prediction using, on the side of the structural information, the other information that we have, the ligand based, the target based, in order to make a better and stronger prediction. So in this case, we didn't look at a specific case, a specific disease, but we look at the entire matrix, compounds, drugs, marketed drugs, proteins, and this is a very huge matrix, very sparsely filled, only 1 or 2%. So it's really impossible to use a canonical molecular approach in order to draw all this new connection. So what I did, and it's going to explain you now, I like to call it Netflix and chill, which is a bit weird as a name, but you will understand why. So let's start from the beginning. This matrix is a very sparse matrix, and in order to fill this matrix, we looked at the Netflix challenge. So Netflix tried to do the same thing using a different matrix, so the matrix of users and movies, and it tried to predict how much a new user could like movies that he didn't watch based on the rating that other users did on the movie that they watched, and also on the rating that the same user gave on the movies that he watched. So first of all, changed the way of rating from a specific number to different categories from 1 to 5. And the same thing we did for the biological activity that we tried to predict in the drug target interaction. And we changed it from a concentration, which can be a continuous number, to 5 different categories. After that, we try to increase the interaction, the drug target interaction prediction by using side information. So a different information from all the different, that can describe all the different entities, and the similarities among those informations could be learned by a model and give us a better better prediction. So this is exactly what a dating app try to do with these users. The dating app asks for your age, ask for your characteristics, your interest, and then try to match your profile with other profile that stays the same characteristics. And the same thing we wanted to do with drug and targets. So based on the physicochemical characteristics, trying to suggest couples that might match. So exactly in the same way how Netflix and Tinder try to match 2 persons together based on their availability and on their side information, so their characteristics, we try to match together drug and target using machine learning approaches that took into account the different physicochemical characteristics. So what are those characteristics? We use 3 different classes: the ligand information, target information, interaction pattern. Ligand informations are the canonical fingerprint that describe the chemical structure of the ligand. Target information, and and I show you here. It's called ECFP, or circular fingerprint, and we tested only those the information present in those fingerprint, and we saw that there is a correlation with the biological activity very high, up to 96%. So only this is already giving a lot of information. Then we also added an extra layer, which is the layer of the protein with 5 different information: the amino acid frequency, so how many of the different amino acid is present in each target, or the sequence similarity. So we made a matrix that express the similarity of all those proteins, or the domain presence. So for each protein, we stated the presence or the absence of a specific domain. And the last 2 categories are structure-based informations and are the residues interaction network. So we build a network of the different residues in the binding site and near the binding site. And so the way, the kind of residue that is there, and also which configuration, spatial configuration it has. And we generated 2 different networks, a more dense one and a smaller one. And for the interaction patterns, of course, we use PLIP, and we transform the PLIP information into a mathematical fingerprint that we could use to describe each different entity. Of course, we wanted to see if those interaction pattern had a signal related to the biological activity. So we did a validation of the data set, and we saw that for each category of biological activity, there was a signal higher than 50%. So something we could rely on. That is not random. The highest one is for the lowest lower threshold of activity, because there were more data available. And this is the structure of the algorithm that we used, this deep learning algorithm that is fusing together the 3 different information, and then giving a final output in the form of AUC, in the form of area under the curve, that states the accuracy of the prediction based on the site information that it used. And this is the final result by running the same model on all different combinations of site information. The one on the right of the blue dash line are all the runs that I did by using structural information, structural data. And the very last one, the one under the category with PLIP, are all the runs that I did by using also the third site information, so the most dense one, the interaction pattern, PLIP. And as you can already see, using the structural information and also using the interaction patterns, the ability of predicting the drug target interaction by using deep learning decreased. And I can show you in numbers this. So this is the list of all the different configuration based on the site information used and the different accuracy values. And you can see that those are in order for the accuracy from the most accurate to the least one. And you can see that the one where PLIP has been used are at the very bottom. So using the it looks like using the interaction profile fingerprint and using this very big amount of structural data kind of makes the thing worse. And this is another table showing the same configuration with or without PLIP, and how with PLIP is always getting worse. So even though there is a lot of information inside the structural interaction profile description of the entities that we showed it works when it's when it's carried on in a molecular point of view, in a machine learning setting, it needs way more attention, and it is a model that has to be that should be designed in order to accommodate all this big amount of data. So in conclusion, for the last slide, just to bring together all the things I've said, first, drug repositioning is something that we can use to overcome the problem of the negative trend of drug discovery. And so far, we've been using the easiest way of repurposing drugs, of course. So we need to bring on more techniques. There is space for structure-based drug repositioning to bring more drug target interaction, new drug target interaction on the market. The problem, of course, we know it is the availability of structural data. So we need more structures, crystal structure or models, reliable models that we can use in order to make predictions. And of course, we have to work on the machine learning, on the deep learning, in order to design models that are able to accommodate this very detailed amount of data that we have from the all the different structure-based techniques in order to help the prediction of new drug target interaction from a very big data kind of perspective. So I think I'm very close to the closure, so they're out of time. So I wanted to thank all the people that worked with me during the PhD and made all this work possible. The people at KU Leuven from both the both the group of bioinformatics at ESA department and in the hospital and the platform of research valorization. The people at TU Dresden that helped me working on the structural-based drug repositioning, and also the organizer of this event that invited me and made all this possible, notwithstanding the weird situation we are at the moment that doesn't allow us to be in the same place and enjoy a conference like we were used to be. And thank all of you for the attention.
So thank you, Daniele. We are open to questions. If someone from YouTube, we have one question here from the committee organization. How do you see the new methodologies in machine learning can affect the scenario of new drug target interaction prediction?
So it's a good question. Thank you for the question. It's very hard to say. It definitely depends on the model. I'm quite optimistic. Machine learning and deep learning has a lot of potential, and at the same time, the structural based information are so dense and can help so much. So we just have to find a way to take the best from the two fields and put them together. So we have to be able to find a deep learning model that is able to not get overloaded with all the information that we can get by fusing all the structural data stuff that we can get from both the target and the interaction, and also bringing together the ligand if we want. We could also add extra layers, the signature, the expression, the differential expression of the cells when treated with different drug. There are so many things we can fuse together, and it's only a matter of tuning and finding the right model. I guess I didn't have the time to try different models, so I tried this one because it's the one that my group in Leuven developed and tried on different topics. I went there in order to see if it was possible to use structural data to make this deep learning model working, and we saw that actually it was getting just too heavy. So I'm sure that will be with the study, with the research, it will be the way to take the best, the advantage from both of the sides.
Nice. And the people are also asking about your opinion on how the AlphaFold and the DeepMind algorithms can impact the structural biology in the next years.
Yeah, well, that was really funny. Yesterday I was revising the presentation, and a friend of mine, a colleague of mine, sent me the link. I was not on Twitter yesterday because I was busy with the presentation, and I started reading. It's like, okay, that's someone wanted to speak about this at the conference for sure, because it was the best moment and the best place to have that news. I don't think this is the death of structural biology. Someone said it's like we are not gonna completely substitute the structural biology and the crystallography with the machine learning. But this is a very strong signal. It says we can learn from what we have, and we are able to make it almost as good as the resolved structure in crystallography. So I'm very happy and extremely optimistic that it's gonna push further the tools that we already have, and it's gonna make the things better. But I don't think that from tomorrow we are gonna use only machine learning to solve all the GPCR structure that we haven't been able to crystallize so far. But definitely something we have to look at with more attention.
Okay, thank you. We also have some comments. Raissa Lima from YouTube, she said that PLIP is awesome, and she used a lot.
Yeah, it is. It's really good, and we've been publishing already multiple articles on PLIP, on how it works, and somehow those interaction profiles, similarities, information are really able to fish good drugs from the pool. So for virtual screening is really good. So I invite everyone to use it because it's a good tool.
So thank you, Daniele.
Thank you.